Hi, the first part didn't record, so you're going to get my editor's play-by-play for this first half. So I started my presentation by thanking everyone for being there and my committee in particular. Um, I made a point of telling everyone that this is me just showing you the best parts of my dissertation, and you need to read the whole thing to find out everything that's wrong with it. And then I basically said my dissertation is in 5 chapters, an intro and conclusion, and then 3 data chapters in the middle. And you could follow along on the bottom there to figure out where we are in the presentation. This is really fun to do. I feel like I should be giving like editor's notes or something. What on earth was I saying during this? It's ridiculous. Probably something really witty though. There's no way to know what I was really saying. Maybe I was wondering why everybody was saying they couldn't hear me on the WebEx. It's quite possible. So I think I'm about to start the real part now. So my broad research question that I'm interested in is not just mercury, as all of you guys know, but even more broad than that, uniting what I did for my master's is how do we quantify and mitigate the effects of human development on wildlife? And you can think of this as there's lots of different stressors on wildlife populations, but such as climate change, urbanization, habitat fragmentation. But the one that I'm most interested in is mercury as a stressor. So mercury is a naturally occurring element. It's concentrated and moved around by humans. But it's also a neurotoxin, and so it has a variety of lethal and sublethal effects on humans and wildlife, from behavior, physiology, immune, cognition, development, reproduction, and survival. But for the most part, we don't see problems with outright mortality. It's more of these sublethal effects that mercury causes, which makes it hard to study and interesting to study. So there's a variety of sources of mercury to aquatic ecosystems, including any day now here. Local point source pollution, where industry or some other type of pollution has occurred that has dumped mercury right into the water. And then non-point source atmospheric deposition, where mercury can move on prevailing wind patterns and be deposited via rain into different aquatic systems. Each of these are important sources, but the one important thing that you need to know is that mercury must be methylated to become toxic. So this conversion of mercury to methylmercury is not going to be studied in my stuff because I'm not a mercury biogeochemist, but methylation does occur primarily in aquatic habitats, and you need to know that there's a variety of factors that influence methylation rates, and they're all habitat-specific factors. Such as, coming up here any moment, wow, I was really talking about this for a while, huh? Ugh, gosh, what on earth was I saying? Here we go. Methylating habitat characteristics include high DOC, large wet dry cycles, and anoxic conditions. Ugh, I feel like I should have had a glass of wine before I started doing this recording. But anyway, methylation and habitat are really, really important. Sorry, you keep hearing Ben's chain rattle in the background there. He's sitting next to me. So the other important thing that you need to know is that methylmercury biomagnifies as it moves through the aquatic system, starting from aquatic insects to insect-eating fish and fish-eating fish and up into fish-eating wildlife. So these two important things that you need to know for the presentation are that mercury must be methylated to become toxic to fish and wildlife, and habitat mitigates this, which is really important to understand. Then the second is that methylmercury biomagnifies as it moves through the food web, which means a high trophic level means high mercury risk. So this is why we're often worried about ocean fish such as swordfish or tuna, which eat at high trophic levels, causing high mercury exposure to humans and wildlife. But there's this big difference between ecosystems, so I've focused mostly so far on the aquatic ecosystem. But you guys all know I'm a bird biologist, so I'm looking at birds living in the terrestrial ecosystem. And what I've found throughout my studies here is that we kind of live in these two different worlds where the aquatic people talk to the aquatic people and the terrestrial people talk to the terrestrial people. And I was really interested during my dissertation of trying to have feet in both worlds, where I wanted to understand the movement of things between these two ecosystems. Um, so basically, this has a certain name called cross-ecosystem subsidies, and you could think of it as salmon moving in stream to bring marine nutrients into fresh water. You could think of it as leaf litter moving into aquatic systems and providing nutrients. I think of it as contaminants and nutrients moving from the aquatic to the terrestrial ecosystem. So this pathway, starting with aquatic insects, the important thing to understand is that aquatic insects go through a life stage where they actually emerge and leave the aquatic environment and become winged insects in the terrestrial environment. And this creates a pathway of mercury exposure from emergent aquatic insects to riparian songbirds. And this is both positive and negative. It's positive because it brings a variety of nutrients into the terrestrial ecosystem, but it's negative when it moves contaminants such as mercury into that terrestrial ecosystem as well. So mercury causes a variety of impacts on terrestrial species. This was a study that I got to work on when I worked at BRI, looking at mercury exposure in the Carolina wren on the South River in Virginia. And we found that mercury was negatively correlated with nest success. So as you can see in this graph that's going to show up any moment now, as female blood mercury concentration increased, the nest success of the different birds declined. And so I'm interested in 3 different spatial scales in my dissertation. One is at the continental scale, looking at western US and Canada, at the regional scale, and at the local scale, looking at the Willamette Valley in particular.
Testing. How about now? Yay! Okay. So okay, that was the boring stuff anyway. Okay.
Hi, Allison. Sorry,
Okay.
the audio wasn't working.
Oh. And now we can hear everyone else.
Be quiet.
Okay, I think Can you still hear us?
Yes.
Yep. Okay. All right, we're good.
Thank you.
Share screen. Okay, so blah, 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 mercury is methylation, biomagnification. You guys all know that who didn't hear it. So my first chapter is using widespread piscivories as model species. Where is mercury risk highest across the whole continental western United States and Canada? Zooming in on the Willamette Basin, I want to look at riparian songbirds and what habitats show the highest exposure to mercury. And then locally, I want to look at what individual birds are eating. So this first chapter is called Mercury Risk to Avian Piscivores Across Western U.S. and Canada, and it's actually already published in Science of the Total Environment. So I'm really going to go pretty quickly through this. Read the whole thing if you're super interested. As you can see, I had 1 million, very awesome, but 1 million co-authors. And the reason for this is that this was part of the Western North American Mercury Synthesis Project, which was funded by the USGS Powell Center, and it basically took a whole bunch of mercury scientists, put us in a room in Fort Collins, and had us synthesize mercury concentrations and figure out how we could develop ability to look at these across all of Western North America. So scientists were there ranging from water, sediment, fish, and wildlife, and I was in the wildlife group. So mercury in the avian piscivore food web, we kind of showed this simplified version. But really, there's a lot of different avian piscivores that we could be concerned about, and there's a lot of different fish that they could be eating, all of which contribute to how much mercury they get. So how do we quantify risk across such a large geographic area? We decided to choose piscivore model species, and we came up with these 5 that we knew had been studied in a lot of different ecotox studies and occurred broadly across Western US and Canada. So bald eagle, osprey, loons, grebes, belted kingfisher. And what we did is we found range maps of each of the species, truncated them to just the western part of the country, and then we went through databases to look for mercury samples. So we contacted individual researchers, and basically we collected no new data for this whole project. We just used things that were in databases. And now I'm going to put up all the dots that we found for belted kingfisher. There were none. We found no Belgian kingfisher samples in our data. So we kind of had to go back to the drawing board. And we did a little bit better with some of the other species, but what we realized is that a lot of these samples are concentrated in places that we already know that there's a mercury problem, or where there happens to be a researcher studying them in that area. So that's not a really great way to assign risk across the whole area. So we went back to looked at these guys again and realized we needed to use fish instead of the actual piscivore samples, because fish are collected across this whole study region for human health implications. So state and federal agencies monitor mercury in fish so that people don't get harmed. So once we went through and looked at fish and we corrected everything to be fish that these birds could be eating, we could get risk maps across the whole area based on fish. And so you don't have to really worry about this too much. Darker red is going to be at more high risk. And we did this for all of the different species that we were interested in. And then we layered them on top of each other to assign broad risk across the whole area. And it looks something like this. I'm just going to highlight a couple areas. One is the Prairie Pothole in Saskatchewan. We saw the Puget Sound, and we saw a lot of spots in the Great Basin. And overall, when we kind of looked at these mercury hotspots, we found that habitat, again, was causing a lot of differences between them, where in some ways it matters if you have a mercury source, but more importantly, it matters what type of habitat you have and if you have methylating habitat. And more often, it was wetlands, river estuaries, and arid land lakes where we saw the highest mercury concentrations. So how do these different habitat types compare within one river basin? That's kind of moving into my second chapter, which is regional differences in mercury exposure in the Willamette Basin. So the Willamette Valley, for everyone who's not out here right now, is in western Oregon. I wanted to look at the Willamette River, which is important to note that the Willamette runs from south to north, where it empties into Portland. And we're here in Corvallis, where that star is. So I was interested in the different habitats in the Willamette Valley and how they could influence mercury for the reason that we know that there is an EPA Superfund site in the headwaters at the Black Butte Mercury Mine. So this was an area that mining for mercury occurred, and it polluted that whole headwater system. That system flows into the Cottage Grove Reservoir, which is a man-made reservoir. It connects to the Coast Fork Willamette River, where it then flows into the main channel of the Willamette River. And then we also sampled a bunch of the Willamette Valley National Wildlife Refuge complexes. So these guys are throughout the Willamette Basin, but not directly on the Willamette River itself. And throughout this region, there are fish consumption advisories for resident fish for mercury. So we knew that there was a mercury issue, but no one had ever sampled riparian songbirds in this area. So we kind of were doing this project to just be able to understand risk to this whole area. And we knew that the habitats varied considerably, and habitat's important for methylation. So at the mine, these are pretty small headwater streams, fast-flowing cold streams. The Cottage Grove Reservoir is a man-made lake with a lot of wetlands, so those wetlands we were thinking are probably good methylating habitat for mercury. The Willamette Valley National Wildlife Refuge Complex is another series of managed wetlands, or at least that's what we sampled within those regions. And then the main channel Willamette and the backwater Willamette are a really interesting system because the Willamette River historically would have been a series of braided channels moving throughout the valley floor. But we've channelized it in a lot of ways. So most of what we see now are main channel flowing gravel bottom streams where we can sample bugs. But there is a legacy of this braided channel in the alcoves and side channels that still occur. And these have much different water flow where water doesn't move through them continuously. They kind of act more like a lake system. So again, we were interested in how this local point source pollution in the headwaters moves through the different habitats and into the riparian songbirds. Right now, I'm just going to show you the results about the riparian songbirds. So what we did is we went out and captured birds in this net, took blood samples, and a lot of morphometric measurements in 2013 and 2014. For this study, we wanted to sample all the birds that we could find living in the riparian forest. So we kind of found whoever was there, and these were the ones that seemed to occur in high numbers. And what we found were some interesting differences among the different regions that we studied. So You're going to see a lot of graphs like this throughout my talk where I'm showing you results from the models that I run that take into account things like sites and dates that are important but we don't want to show. So here on the y-axis is going to be least squared mean bird mercury concentrations. So at the Black Butte mine, we saw the highest mercury, and moving downstream, we saw lower mercury at the Cottage Grove Reservoir. And when you look at these and you see different letters, that means they were statistically different. All the way downstream in the main channel of the Willamette, we saw significantly lower mercury concentrations, so that's kind of what we were expecting. But in these other kind of different wetlandy habitats throughout the area, we also see elevated mercury. And don't get too distracted by that one. We had really low sample size in that area, and I think that's why the arrow bars are so big. But for the most part, focus on the fact that while the wetlands and the backwater areas have no significant difference from the Cottage Grove Reservoir, which is listed as an EPA Superfund site for mercury contamination. So something's going on here with the habitats that can cause methylation to occur far downstream from where we knew the source was. And also, interestingly, we found a big difference among the species that we sampled, ranging from, at the far end, the black-headed grosbeak had the lowest mercury concentration, and at the highest end. And as you can see from these guys, especially the grosbeak gnawing on my finger, they have very different foraging ecologies. We know that black grosbeaks are more omnivorous, they'll eat seeds and fruits. Buextrons are pretty insectivorous. And then towards the middle of the mercury exposure profile was the song sparrow, which we think of as more of a generalist. And for all the bird people in the room, when you look at this, you can kind of see this progression from something that we would assume would eat a lot of plant material to the ones that are more insectivorous. So the conclusions from this chapter are that habitat matters. We saw differences within the different regions. And species are not all created equal. We saw big differences among the species. So why does mercury vary so much among these songbird species? Which leads me into my last chapter. I'm going to talk about pulse reliance on aquatic carbon subsidies influences mercury exposure in riparian songbirds. So I showed you this riparian food web that's fairly simple. So aquatic insects emerge, become emergent insects, and are eaten by riparian songbirds. But there's actually a lot more going on because there's this whole piece of terrestrial productivity where terrestrial insects like caterpillars can feed on entirely terrestrial matter, and then we can see terrestrial invert predators like spiders also feeding on either emergent insects or terrestrial insects, and then songbirds can choose to eat any combination of these that they want. So they forage across whatever they can find based on their own preferences. So for this study, I zoomed into just the upper and middle Willamette River. My farthest south site was Irish Bend, and my farthest north site was Grand Island, which is north of Salem. And for this study, I focused on common riparian songbirds that I could catch at each site so that we'd have replication across sites. And the objectives were to look at what factors influence mercury, how much aquatic carbon do individual riparian birds eat, and does this correlate with their mercury exposure? So for this first part, I wanted to think about what different things could influence mercury exposure in these guys. The first one was that we have vastly different habitat types in the Willamette. So again, I'm going to look at the difference between main channel and backwater habitats. And then second, I wanted to look at time of the year. So for those of you who are in the room or listening who are there for that 2013 field season, it was a long field season. We started around mid-April and went to the end of July. And I truncated these to be what we considered breeding birds before they molted. So I made sure they were resident in this area and hadn't started molting. But this actually spans a large part of their breeding season, from early breeding attempts to maybe mid-June, which is like, maybe they're re-nesting or doing a second nest. And then finally, through their late fledging period where they're caring for their young. And in the Willamette, we see a big shift in weather between these two seasons, where early in the season it's super rainy and wet, and then it turns into what we hope is coming soon, the sunny Willamette summer. And then I use species as a proxy to look at their foraging preferences, because I picked these 5 species, and we kind of consider Swainson's thrush, song sparrow, and towhees as more generalists and warblers and yellowthroats as kind of insect-eating birds. So I basically built a big mixed effects model that looked at these three factors and all two-way interactions and included random effects of sight to account for potential differences among sites. And one of the big findings from this model was that backwaters have higher mercury than the main channel across species and across dates. So no matter what time of year you were looking at, we always saw elevated mercury in the backwater areas. And so we kind of are coming back to that idea that habitat matters for mercury methylation. And this was kind of an interesting part of it, that we also found that species mercury varies by date. So early in the season, we see all the species kind of similar, but they have different trends throughout the season, where by the end of the season, we see that Swainson's thrush have declined considerably, making them different in mercury concentration than the other birds. Overall, we generally see a downward trend in most of the species that we studied, where their mercury is highest early in the season and decreases later. And then the yellow warbler, we kind of saw no trends, so they seem to be staying consistent. And I'm not going to show you, but I ran similar models for aquatic and terrestrial inverts, and the results were that we found that throughout the same time, aquatic bug mercury increased, terrestrial bug mercury stayed the same and super low throughout the season. But I just showed you that bird mercury declines throughout the season. So how is this possible? How are we seeing an increase in aquatic mercury but a decrease in bird mercury? And this brings me to my last two objectives, which was looking at how much aquatic carbon do individual riparian birds eat, and does this correlate with their mercury exposure? So what we did is, when we were out sampling birds, we also sampled aquatic and terrestrial invertebrates at each site. And what I wanted to do was use isotopes to figure out where did the carbon in the tissue come from. So we can use the delta-13 carbon isotope to assign that. What you want is a difference between your aquatic signal and your terrestrial signal. And we use these primary consumers in both compartments, hoping that there would be a difference between these two. So full disclosure, one of the things, it's not that it didn't work, it's that the backwater sites have a really complicated carbon isotope signal, so I'm not going to show you guys any of those results, but it's really interesting. It just would take the whole time to explain it. So what I'm going to show you is instead just the results from the main channel, where we got this really good differentiation between aquatic and terrestrial prey. And what this is going to look like is an isotope plot where carbon is on the x-axis and nitrogen is on the y-axis. Carbon is going to tell us the carbon source, if it's aquatic or terrestrial, and nitrogen should show us trophic position. But for people who are into isotopes, I'm not going to end up using nitrogen because I had a difference in baseline between the different habitats that I was looking at, and it didn't work out that well. So what these things end up looking like is we want there to be a difference, right, between aquatic and terrestrial, and we found that. So we found that in carbon, there's a big difference between aquatic on the terrestrial on the left and aquatic on the right. So you can think of this as anything to the right is going to be more 100% aquatic, anything to the left is 100% terrestrial. But where do something like a spider fit into this whole thing? If we plot now predators over this, because so these two end members are the primary consumers, if we plot predators, we now see this interesting pattern where the blue dots are aquatic predators, so dragonfly larvae, beetles, things that are in the water, and the green dots are terrestrial predators, invertebrate predators like spiders, harvestmen. And so we see, for the most part, the aquatic predators map over the aquatic carbon signal. It seems like most of our spider predators are down here feeding slightly aquatically and slightly terrestrially. So what's the deal with this dot up here? I find it super interesting. This is a tetranathid spider, so these are a type of spider that are known to be fishing spiders, so they live only in riparian zones and feed primarily on emergent aquatic insects. So it makes a lot of sense that they would be tracking right over the aquatic signal. And luckily for us, in the main stem sites, the birds also showed a difference between feeding 100% terrestrially and 100% aquatically. So we kind of see this differentiation between them. And we can look at this a different way in terms of just looking at the carbon isotope. And we see differences in mean carbon isotopes among the different species. And now you're going to think, Farther up is going to be more aquatically feeding, farther down is more terrestrially feeding. And what I ended up doing was using a simple 200-member mixing model for isotopes to assign for each bird the proportion of aquatic carbon that was in the sample that we tested. So what we found, which was really interesting, was that birds eat less aquatic carbon later in the season. So again, we did a model that accounted for site differences, and we found consistently across that birds are declining in the amount of aquatic carbon that they're eating throughout the season. This does vary, the species do vary in the amount overall of aquatic carbon that they're eating, but overall they all decline throughout the season. So could this explain the individual differences that we're seeing in mercury? We see that aquatic carbon is declining throughout the season, and we see that bird mercury is declining throughout the season. So how does aquatic carbon relate to mercury? We again wanted to look at this correlation between proportion of aquatic carbon and mercury. This is going to be a modeled result that I'm putting up that accounts for different baselines mercury between each site. So now we're taking into account if it was a backwater site or a main channel site. Oh, no, these are all main channels, sorry. We're accounting for the differences between the actual sites. And we see a very strong correlation between the proportion of aquatic carbon and their mercury exposure. But we see that the birds kind of vary in their baseline of how much mercury they have in their sample. And they kind of vary in how generalist they are. So those brown dots are the song sparrows, and we see them spanning almost the whole range from low aquatic carbon to high aquatic carbon. So what does this all mean for Willamette birds? Is that clock actually right? Yeah, okay, cool. So I showed you this graph that showed how mercury declined on the South River. The highest sample that we found in the Willamette Valley is still at the very low end of contamination that we saw in Virginia, causing this level of effect. And most of our birds were in this more low range, which on the South River we actually would have called this reference or uncontaminated. So what does this mean for the birds in our area? I would like to think that it probably means that mercury isn't the biggest stressor on the bird populations here. It's at relatively low levels. But we did sample birds like the Willis flycatcher that are declining in a lot of ways, and they did have some of the higher mercury concentrations that we found. So I think this is where we have to think of mercury as one of many stressors that could be on bird populations. So it's not that mercury is causing these declines, but maybe once you add in a few other stressors, we could see issues in their reproduction. So we found that individuals vary in what they eat and their mercury exposure. So bird mercury declines throughout the season. And this has important implications for bird reproduction and mercury monitoring, because depending on what time of the year you went out and sampled birds, you would get very different results for how high their mercury exposure was. And if we think about this just from an assessment standpoint, that matters, because if you're going out to assess if mercury is a risk, you want to make sure you're capturing potentially the high risk. But it also matters for the birds, because early in the season, especially here, that's when they're trying to breed, trying to get their first clutch off the ground. It's also when they're dealing with kind of bad weather and all of that. So this could be a very stressful time for birds, and it's also when they have higher mercury exposure. So maybe not in the Willamette, but I believe these processes are probably similar in other sites that have more mercury contamination. But you have to be careful about that. You want to be worried about mercury exposure during the time that's most stressful for the birds. And future directions, we saw this decline in bird mercury and this decline in the amount of aquatic carbon eaten, but we didn't have any actual idea about the flux of aquatic insects coming out of the terrestrial ecosystem. So what I think would be really cool is now to kind of monitor some of that, where we can actually get an estimate of how much biomass is leaving the aquatic system and how much mercury it's bringing with it into the terrestrial ecosystem. So the conclusions of my dissertation are, you are what, when, where, and how you eat. So what does that mean? You are what you eat because it's the difference between aquatic and terrestrial carbon seems to be really important for mercury exposure. You are when you eat because early or late in the season makes a big difference. You are where you eat because we see all these differences in habitats across the area and mercury methylation. And you are how you eat, which is pretty much unanswered in a lot of parts of my dissertation, but we see different baselines in a lot of these species that could be assumed to be with what they're actually eating. So I said that we couldn't deal with nitrogen in this. It would have been really great to get trophic position, because maybe some of these birds that have higher mercury exposure are also feeding a couple trophic levels up by eating primarily spiders that are eating maybe something else that's eating aquatic insects. So there's a lot of unanswered questions here about species foraging preferences. So why does any of this matter? And I took out the part that says maybe it all doesn't, because that was last week Allison, who was pretty, pretty stressed out. It does matter. It matters from a mercury monitoring and assessment standpoint. You know, mercury is something that is monitored by all sorts of agencies, because different industries have polluted, and those industries should pay for restoration attempts. So we want to know what the mercury is in these different end members so that we can get a good assessment of what's going on. In terms of riparian bird ecology and conservation, I found interesting things that, you know, I didn't potentially mean to find, but in that these birds are changing really dramatically the source of carbon that they're eating throughout the season. And this also means that when we're studying riparian bird ecology, we need to be kind of aware of that to know that these birds are probably shifting their diet more so than maybe we originally thought. And then in terms of and restoration, you know, we're often restoring habitats for fish. I've been to a lot of fish talks about stuff like that. But you can also maybe use some riparian birds in some of your analyses, because when you're restoring a stream that increases emergent prey, those prey, some of them are going to survive the fish prey and emerge out into the ecosystem and help provide a prey source to riparian songbirds. So we should probably all be talking a little bit more about how we can say that our projects are good for more than one end member. So I want to thank all the OSU undergrads, like some of who are here. It's really amazing. So my entire field project was done by me and interns that came and helped me, which was really, really amazing. And they endured really early mornings and really horrible field conditions at certain times. And this, you know, wouldn't have been possible without getting them out there. And it really helped me figure out that, you know, I want to be a teacher in the end, so it kind of helped shape my career path. I have a lot of people to thank. I don't want to keep you guys here forever. Obviously, Ben the dog is really important. And everyone else who's kind of helped support me through this, whether in funding or, you know, reading over manuscripts or helping me kind of relax and get through this whole process. I really, really appreciate everyone's help. And I'll leave that up so that you can all see your pictures, and I'll take any questions. Thank you.
Plenty of time for questions, so.
Dan. Yeah. So the question was, did I sample near the Superfund site? Yeah, that Black Butte headwater, I was in the Superfund site, part of which has been remediated. So we didn't see a big difference because I think the birds are kind of moving around that whole area. But that's the part that was highest, the Black Butte mine. Yeah, no, we did. Yeah, the Black Butte is basically the Superfund site. And the Cottage Grove reservoir is technically considered a Superfund site, and we sampled all around that too.
And that pattern of fire, debris, fire.
Oh, so we didn't do that study in, the headwater area, because it's a very small area, and the headwater streams are different than the Willamette, so we focused that part on the big parts of the Willamette that we could catch enough birds to do a study like that. I think it would be different up there. The emergence would be really different, but because we know that the birds are getting exposed, it's definitely probably a similar pathway where it's coming out that way. Scott?
You measured your mercury in blood.
Mm-hmm.
You measure mercury in different tissue compartments, and you've seen something different than others. These birds are coming in to this area from somewhere else, migratory.
Yeah.
And so what sort of legacy mercury might they be bringing in?
Yeah, that's a good question. So the question was about if we sampled in another compartment, a different tissue, what we find different things, and they are migratory. So I looked at this because some of our birds are resident, some of them are migratory. So the blood, we tried to make sure we were sampling birds that their blood signal would be about the last month, and that was the month that they were in the habitat that we sampled them in. It's a good question. I hope the undergrads are listening about feathers. So my undergrads who worked with me worked all on looking at feather mercury because it's really complicated because they grow their feathers the year before we were catching them, and they usually grow them for these guys maybe on a different site, but what I think is happening is they're eating very different food when they're growing their feathers because it's post-breeding season. They're probably eating a lot of vegetable matter at that point. So we see a terrible connection between blood mercury and feather mercury, and even a pretty bad correlation between liver mercury and feather mercury. So feathers are, I just don't think, a good end member when you're trying to get at this. But, I didn't want to kill any of the birds, so I couldn't do like a muscle or a liver sample, but those kind of show a longer time period, so those would be bringing in some of the old mercury that they've experienced throughout their life cycle. Does that answer your question? Yeah.
I have a serious question.
Serious.
Serious question. So do you think that maybe doing something like a stomach analysis or would kind of elucidate more of the question of what birds are eating when? Could you do something like that to inform the study a little bit more?
Yeah. Yeah. Like killing the bird?
No.
Jenna said it. No. I don't think you can do a gut content analysis on these guys in the same way because they process through their food so fast when they're breeding, their throughput is really, really fast. So a gut content is going to be what they ate that day, and we kind of want a longer signal maybe. I would really like to get, maybe getting the idea of the prey samples that are available to them to eat would be really interesting, and we didn't have time to do that. And then you could do the type of thing where you can get what the parents are breeding to their young. That would be interesting, and that would kind of all get around it. But I don't think there's any real reason to do a gut content study. What was the fun question?
Experience with the mercury analysis.
Oh God. Which one? Yeah, no, the mercury analyzer was the worst. These samples, I think, we got good results in the end. It just didn't work on the timetables that I always wanted it to. It seemed to go down a lot. And I'm not asking for a mercury analyzer at my future job because I don't want to deal with any of those issues. Because it's just not fun to have that stress. Colin deals with all of it for our lab. Yvonne?
Was there any role of the lifespan of different species in the population?
Probably. Lifespan of the birds or of the inverts? Yeah. So most of these birds' lifespan, are pretty similar. They're pretty, short-lived for some bird species comparatively. I think it could, in terms of what mercury effect has, I think their ecology can play into that because some of these are long-distance migrants where they go down to South America, which is a very taxing thing for them to do. And so I think mercury could come into play when you're dealing with something like that, when you have to migrate. So I'm not sure if it's life expectancy as much as the different things that the species go through in their life cycle. Mike?
I have a couple of questions. Do you have a sense of how well the decline in the aquatic carbon input aligns with the technology insects?
Yeah, I would assume it does. I don't have any data on it. We couldn't find any good information on emergence in the Willamette besides from fly fishing guides, where they do think, the idea is that emergence is higher early in the season where we saw that high pulse of aquatic carbon. And from experience, we know that, once it dries out and the terrestrial productivity gets pumping, we saw a lot more of terrestrial prey available. But it's all kind of anecdotal because we didn't go out and do the actual sampling to see what was available to them.
I'm also curious if you had any feedback. If you did, how much variation?
Yeah. That sounds like a loaded question because he was on my field crew. But we didn't get hardly any, and I excluded, I think there were 2 in 2013 that we got recaptured within the season. And that was surprising. I kind of went to a lot of the same sites again, hoping to recapture the same birds, and I didn't find them again. And of course, I was worried that they had died. But we found more recaptures between years. So when we went back to a site in 2014, the birds had come back to their same spot, and they were there the next year. But something was going on, and it's probably something having to do with the birds nest, right, early in the season by the riparian area. If they're successful, they probably move somewhere to take care of their fledglings, and a new group comes in, and those are the ones that had this terrestrial carbon signal. So it could be that, really complicated, it could be that those birds move from a more terrestrial area in, and that's why we're seeing it. That's why we kind of need to get the flux of the two to see what's happening.
I guess I was asking about the mercury.
Oh yeah, so mercury shouldn't vary within the season because we didn't include any birds that had started molting, and that's really the only pathway that they have to excrete mercury throughout the season. So on our recaptures, we really didn't see any difference in mercury exposure between them. So even if they had we did see like a couple that were eating different prey, like we saw that they had changed their carbon signal, but their mercury didn't change because at that point they had nowhere for the mercury to go that they had already eaten a month before. Thanks.
I'm wondering about whether we're doing people storehouse or something other.
Yeah, and there have been studies of that. It's really cool. You can usually just use feathers because when they grow their feather, they bind the mercury into it and it's completely inert. But a lot of museum specimens have been prepped with methylmercury, or with inorganic mercury, I'm sorry. They're prepped with inorganic mercury, so you basically bonk the mercury analyzer if you try to put that through. So we can do it. You have to run them for methylmercury because that's not what they were prepped with. And you can see, we did a study when I worked at BRI on songbirds, like throughout time. We didn't see a great, huge decline in that, but we didn't have a lot of samples like pre-industry. But they've done it in like seabirds, and they see a really pretty significant spike of mercury after industry started polluting.
I'm worried if it's on the effect of gender on the changing mercury tests, in particular, like if females exclude mercury.
Yeah. So I checked for differences in sex, and I didn't find any differences, and I was kind of worried that I didn't have a lot of females in my data set, because for the most part I was using active capture, so I was playing songs and catching mostly males who were territorial. So I was worried that finding that there's no difference isn't exactly true. You'd really have to do a paired study between the two, the male and the female in the same territory, to see if there was a difference. Because you're right, like birds, their other pathway to excrete mercury is through egg laying, so females could have lower. In some of the bigger data sets that I've worked on, I don't see huge differences between sexes when you're looking at like just a site-specific basis. We saw it in Carolina wrens, where we had both the male and the female in the same territory, but I think that could have more to do with like energetics of the birds, that the males are during breeding, and so they might be burning through calories and eating more high-order prey and getting higher mercury. So I'm not sure that egg laying actually is a significant separation point for birds, at least for songbirds. All right, we're going to squirrels tonight. Thank you.